physicists at the University of Göttingen, together with pathologists and lung specialists at the Medical University of Hannover, have developed a three-dimensional imaging technique that enables high-resolution and three-dimensional representation of damaged lung tissue following severe COVID-19. Using a special X-ray microscopy technique, they were able to image changes caused by the coronavirus in the structure of alveoli, the tiny air sacs in the lung, and the vasculature. The results of the study were published in the research journal eLife. In severe COVID-19 disease, the researchers observed significant changes in the vasculature, inflammation, blood clots and hyaline membranes, which are composed of proteins and dead cells deposited on the alveolar walls, which make gas exchange difficult or impossible. With their new imaging approach, these changes can be visualized for the first time in larger tissue volumes, without cutting and staining or damaging the tissue as in conventional histology. It is particularly well suited for tracing small blood vessels and their branches in 3 dimensions, localizing cells of the immune systems which are recruited to the inflammation sites, and measuring the thickness of the alveolar walls. Due to the 3-dimensional reconstruction, the data could also be used to simulate gas exchange. Using zoom tomography, large areas of lung tissue embedded in wax can be scanned, enabling detailed examination to locate particularly interesting areas around inflammation, blood vessels or bronchial tubes, says lead author Professor Tim Saldit from the Institute of X-ray Physics at the University. Since X-rays penetrate deep into tissue, this enables scientists to understand the relation between the microscopic tissue structure and the larger functional architecture of an organ. This is important, for example, to visualize the tree of blood vessels down to the smallest capillaries. The authors foresee that this new X-ray technique will be an extension to traditional histology and histopathology, areas of study which go back to the 19th century when optical microscopes had just become available and pathologists could thereby unravel the microscopic origins of many diseases. Even today, pathologists still follow the same basic steps to prepare and investigate tissue: chemical fixation, slicing, staining and microscopy. This traditional approach, however, is not sufficient if 3-dimensional images are required or if large volumes have to be screened, digitalized or analyzed with computer programs. 3-dimensional imaging is well known from medical computerized tomography (CT). However, the resolution and contrast of this conventional technique are not sufficient to detect the tissue structure with cellular or subcellular resolution. Therefore, the authors used phase contrast, which exploits the different propagation velocities of X-rays in tissue to generate an intensity pattern on the detector. Saldit and his research group at the Institute for X-ray Physics developed special illumination optics and algorithms to reconstruct sharp images from these patterns, an approach which they have now adapted for the study of lung tissue affected by severe progression of COVID-19. The Göttingen team could record lung tissue at scalable size and resolution, yielding both larger overviews and close-up reconstructions. Depending on the setting, their method can even yield structural details below the resolution of conventional light microscopy. To achieve this, the researchers used highly powerful X-ray radiation generated at the PETRAII storage ring of the German electron synchrotron (DESY) in Hamburg. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you new tech videos. Thank you.